In a particular nucleotide, there are two bonds. We learned about the first bond, that is an N-glycosidic linkage, which is formed between a nitrogen base and a pentose sugar. Whereas the phosphate group is attached to the fifth carbon atom of the pentose sugar by means of another bond, which is called as the phosphoester bond. So two bonds are present, the N-glycosidic linkage and the phosphoester bond. The two nucleotides are linked together to form a dinucleotide. Two nucleotides are linked together to form a dinucleotide, and these are joined together by means of a bond called as 3-5-phosphodiester bond. And let us see how this phosphodiester bond is formed. We learned just now that a phosphate group is attached to the fifth carbon atom by means of a phosphoester bond. And in this figure, we can see that in the first nucleotide, which is having an adenine attached to the first carbon atom, and to the third carbon atom, we can see a phosphate group which is attached over here at the third carbon atom. And this phosphate group is again attached to the second nucleotide at the fifth carbon atom of the second nucleotide's pentose sugar. So the bond is formed between the third carbon atom and the fifth carbon atom of two consecutive nucleotides pentose sugar. So that's why it is called as 3-5-. And how many ester bonds are formed? Two ester bonds are formed. Okay, so it's called as 3-5- phosphodiester bond. And it's very important to know that in RNA, each nucleotide has an additional OH group. When compared to DNA, the RNA is having an OH group, that is a hydroxyl group is present at the second position, or the second carbon atom is having an hydroxyl group attached to it. Whereas this hydroxyl group is absent, or the oxygen is absent in the case of DNA, and hence the name deoxy. Deoxy, that means the ribose sugar is lacking an oxygen, whereas it is present in RNA, and this oxygen is absent in DNA. So the RNA is having an additional OH, or the hydroxyl group, at the second carbon atom in the ribose sugar. About the discovery of DNA, it was Friedrich Miescher in the year 1869 who identified DNA, and he gave the name as nuclein. So Friedrich Miescher gave the name nuclein, and the structure of the DNA was elucidated by James Watson and Francis Crick. It was these two scientists who proposed the structure of DNA, and they told that it is a double helix model of DNA. It's a double helix means a spiral ladder, and we can see the structure of DNA. This is a double helix model of DNA. And they proposed this double helix structure of DNA based on the X-ray diffraction studies, which was proposed by Maurice Wilkins and Rosalind Franklin. So the two scientists, Maurice Wilson and Rosalind Franklin, they had proposed the X-ray, they had conducted X-ray diffraction studies. And based on these X-ray diffraction studies, Watson and Crick proposed the double helix model of DNA. The structure of DNA as proposed by Watson and Crick, it's a double helix model. So how many polynucleotide chains are present in DNA? There are two polynucleotide chains which is present in DNA, and they are coiled around each other. They are coiled around each other in a right-handed fashion, that is towards the right-hand side. And the pitch of the helix, what do you mean pitch? The turn that the helix makes. The pitch of the helix is 3.4 nanometers, or it is 34 Armstrongs. So the pitch of the helix is 3.4 nanometers. And in each turn, the number of base pairs is 10. What do you mean by base pair? The nitrogen bases on one polynucleotide is complementary to the nitrogen bases on the other nucleotide chain. We will learn that in detail in the following slides. So the pitch of the helix is 3.4 nanometers, and in each turn that the helix makes, there are 10 base pairs, and the distance between each base pair is 0.34 nanometers, or it is 3.4 angstroms. So clear? So the distance between each base pair is 3.4 angstrom, or it is 0.34 nanometers. The backbone of DNA helix is made up of the sugar and the phosphate. In this picture, we can see what is the backbone made up of. This is a single polynucleotide chain, and the backbone is made up of sugar and phosphate. So the backbone of a polynucleotide chain is made up of sugar and phosphate, and the bases project inside. We can see this is one polynucleotide chain, and this is the other polynucleotide chain whose backbone is made up of sugar and phosphate, and we can see the bases projecting inward to the helix, and the two chains, they are running in an anti-parallel direction. They are running in an anti-parallel direction. I will tell you what is an anti-parallel direction. In this picture, we can see that at one end of a polynucleotide chain, there is a phosphate moiety or a phosphate molecule. And at what position is it present? There is a free phosphate molecule at the phi dash end. At the phi dash end of the polynucleotide chain, and this end is called as the phi dash end. Whereas at the other end, there is a free hydroxyl moiety or a hydroxyl molecule at the 3' end. So this end is called as a 3' end, and this is called as the phi dash end. So this is the direction in which a polynucleotide or a DNA strand is running, that is in the 3' to phi dash direction. In this case, we can see that this is running in the phi dash to 3' direction, whereas the other strand is running in an opposite direction, that is from 3' to phi dash. That means they run anti-parallel to each other. That means their polarity is in the opposite direction. If one strand is running in the 3' phi dash direction, the other strand will run in the phi dash to 3' direction. So the backbone of DNA is made by the phosphate and sugar components, whereas the nitrogen bases project inward, and the two strands run in anti-parallel direction.